today starting a new chapter, which is GIT. So GIT. So one of the common questions that you get is the first one: name the salivatory gland composition, function, and regulation of saliva. So this question is quite easy. You could do the reading and then do your notes yourself, or maybe I'll upload the notes, but for today we'll concentrate on gastric juice. So mainly the gastric secretion, which consists of the glands and then the cells which make up the glands, and then the secretions which are secreted from the cells. And then we'll talk about the composition of the gastric juice, the mechanism of secretion of HCl, regulation of HCl, and finally phases of gastric secretion and inhibition of gastric secretion. So first of all, we'll talk about gastric secretion. So as we said, first you'll have the glands. So there are mainly two types of glands in the stomach. So first of all, you'll have the oxyntic gland. It makes up approximately 80% of the stomach. So as you can see here, the proximal 80% of the stomach, it is made up of oxyntic gland, and then the distal 20% of the stomach is made out of pyloric gland. So let's focus on the oxyntic gland first. So the oxyntic gland consists of three types of cells. So as I said before, cells make up glands. So there are three types of cells. Firstly, you will have mucous neck cells which form mucus, and then you have peptic cells which form pepsinogen, and then oxyntic, or more commonly known as parietal cells, which form HCl and intrinsic factor. So keep this in mind. We are going to discuss about this more later. And then we'll have the pyloric glands, which are found at the pyloric end. So for the pyloric gland, as you can see, the first two are the same. So you'll have again mucous neck cells which form mucus, peptic cells which form pepsinogen, and what's different is G cells which release gastrin. In comparison, here you'll have parietal cell which will secrete HCl. So that's it for the glands and cells. So here you'll see a histological view of the stomach. Here in green you'll see the parietal cells, you'll see the neck mucus cells, and then chief cells and all. So let's move on to the first one. You have completed it. Okay, so now let's move on to the composition of gastric juice. So very easy. The gastric juice, so first of all, the daily secretion will be from 1.5 to 2.5 liters. pH, so it's very acidic, 1 to 2, and then it's secreted by gastric gland, pyloric gland, and surface mucus cells. So the composition, so 99.5% it's made up of water. In some books it's even written 99, but like it's negligible. And then the other 0.5% it's made up of inorganic substance and organic substance. So for inorganic substance, we will have HCl, a very important constituent. And then you'll have different ions. So we'll have potassium, calcium, magnesium, and sodium. And then we'll have phosphate molecule. You'll have sulfate molecule, and it's how's it called? Forgot. Okay, so as for the organic substances, we have enzymes. Which are mainly gastric lipase, pepsin, which is the active form of pepsinogen. We have gelatinase and amylase, and then we also have intrinsic factors which are very important for the absorption of vitamin B12. Okay, so that's it for the composition. Done. And then for the one of the more important ones, the mechanism of HCl, which is very easy. So here it is. It might look very daunting, but if you do it stepwise, it will be easy. So the first step, we'll have carbon dioxide from the, so this is a capillary. Here it's a capillary which is related to the stomach. So this is a capillary. There will be transport of carbon dioxide. So first let's have some perspective. So this is a capillary. This will be your parietal cell, which is this cell which forms HCl. Earlier we discussed it. Here the parietal cell or the oxyntic cell. So and here they are in histological diagram, as you can see. So this parietal cell, what happens inside? Here you'll have the capillary, the parietal cell, and then you'll have here the lumen of the canaliculi. So first of all, you'll have transport of carbon dioxide into the parietal cell. What this carbon dioxide will do, it will react, so it will be hydrolyzed by water in the presence of carbonic anhydrase, which will form carbonic acid. So this one is called carbonic acid. And then this carbonic acid, it will dissociate into carbonate and H plus. So carbonate ion and H plus ion. So what does this H plus do? Well, here we'll have the first important transporter, which is the hydrogen potassium ATPase pump or transporter. So it will carry out H plus to the outside into the lumen of the canaliculi, and then it will bring in potassium inside. So we have talked about this. So now for the carbonate ion. So the carbonate ion it will be basically excreted out of the parietal cell into the capillary in exchange for chloride ion. Okay, and then we'll have, as you can see, there's potassium, there's efflux of potassium, and hence there will be influx of sodium in order to keep up with the electrical charge, to maintain a balanced electrical charge. So what will this Na do? It will come, and then again it will be transported into the capillary in exchange for potassium, and then the potassium will be secreted out into the lumen of the caniculi. So first we have talked about this, and then the second we have talked about this, and then the third one is sodium potassium ATPase pump. Okay, and then as you can see, chloride here, it moves down its electrochemical gradient into the lumen of canaliculi, and then oxygen is transported into the lumen through osmosis. So why did we do all of this? So the only reason is we need H plus and Cl minus to form HCl. Okay. So H plus, we have H plus in the lumen, and the concentration will be 155 milli equimolar per liter. So here we'll have 150 milli equivalent per liter. Okay, and then calcium will be available at 173 milli equivalent per liter. So as you can see here, H plus is the limiting agent. So what will be the final product of all of this? We will have, first of all, mainly we will have water. Okay, we will have HCl, as you can see, at 155 milli equivalent per liter, because as we saw here, HCl is the limiting reagent. And then we will have a small quantity of NaCl, because Na will react with the Cl which is in excess. So this sums up the mechanism of HCl secretion. So we are done with this. So I'll advise you to draw this diagram like several times, and then you'll get the hang of it. Just remember the sequence, and then it will be easy on you. Okay, and then we'll talk about lastly the regulation of HCl secretion. So HCl secretion will have yes, so we'll have, first of all, the action of the vagus nerve, and then we'll have here, it's called the G cells, and then here ECL, which stands for enterochromaffin like cells. So let me write it. So enterochromaffin like cells. Okay, so what will happen here? First of all, let's talk about the vagus nerve. Upon stimulation, the vagus nerve will form acetyl-CoA, but it will also stimulate the mucus cells, which will form mucus, and then peptic cells, which will form pepsinogen. So again, we will have the stimulation of vagus nerve, which will form the which will stimulate the formation of acetylcholine. So what will acetylcholine do? It will activate this receptor, which is called the M3 receptor. What will this receptor do? It will, so basically it will increase the concentration of calcium. So here, first of all, let's talk about here. What is this? It's the parietal cell. It's the same cell that we have been talking about. So there's basically two pumps which are like the main controlling pumps, which are the hydrogen potassium ATPase pump, which is seen here, and then the chlorine pump. Okay, so if we can stop the action of either one of them or increase the action of either one of them, we'll able to increase or decrease the secretion of HCl. So here we are talking about increasing the secretion of HCl. So let's get back to it. I said vagus nerve, it will form acetyl-CoA, and then this acetyl-CoA will act on this receptor, which will form Ca2+. So let's keep this in mind, and then we'll have this vagus nerve. Upon stimulation, it will also stimulate G cells. These G cells, what they do? They increase the secretion of gastrin. And what does this gastrin do? Again, it will act on specific cell, which is called the CCKB2. So you'll have to learn this by heart. CCKB2, and what will this do? Again, the secretion of calcium. And then this gastrin will in turn stimulate the enterochromaffin like cells. It will release histamine, which will act on H2 cell, which will convert ATP into, so basically ATP into cyclic AMP. Okay, so we have talked about calcium and cyclic AMP. So why do we need these? So calcium and cyclic AMP, as you can see, they will stimulate protein kinase, which here in this case it's very, very important. Why? Because it stimulates this hydrogen potassium ATPase pump. And what does this do? It will increase the secretion of H plus. Okay, and what did we say here? H plus, it is the rate limiting reagent. So as we increase the secretion of H plus, we will increase the secretion of HCl. So I hope this is clear. And then we'll talk about for people who have high HCl secretion, there's a drug called atropine, which will block the M3 receptor, and then it will decrease the action of M3 while still maintaining the action of CCKB2 and H2. This will slightly decrease the excretion of HCl. So let's have a recap. We have talked about gastric secretion, composition, mechanism, and lastly regulation of HCl. And as far as these two topics. They're quite easy, so like just go through them and try to make your own notes, or well, yeah, like try to make your own notes first, and then maybe if I have time I'll do the notes on these two. And then there's another thing, read on this also, gastric histamine reflex. So this sums up the video. So just go for it and then do some reading in the book, and then hopefully you'll have the growth.